Look closely at a clownfish group, and you will find a very specific arrangement. The largest fish is the breeding female. Her partner is smaller, and the rest of the fish are smaller still. Now remove the leader. In many clownfish groups, the second largest fish does not swim away and wait for a replacement. He starts moving into her position and over time becomes the new breeding female. That headline is real, but it hides the interesting part. This is not an instant costume change. It is a social signal that unfolds through the brain, hormones, and gonads. These fish are anemonefishes, clownfish that live in close association with a host sea anemone. In the well-studied species Amphiprion percula, a group can contain a breeding pair and 0 to 4 non-breeders. Their rank is strongly tied to size. The largest fish is rank 1, the breeding female. The second largest is rank 2, the breeding male. Then come progressively smaller subordinates. This is effectively a cue for the breeding position. A fish does not become female simply because it reaches a certain birthday. Its social position determines which reproductive role is available. When the dominant female dies or is removed, the group loses its rank 1 fish. That changes the social environment before it changes the anatomy. The breeding male is now the largest remaining adult, so he ascends. He becomes more dominant, defends the anemone, and begins to occupy the role that used to belong to the female. At the same time, the largest subordinate moves up behind him. This is why the event is better described as a cue advancing than as one fish magically switching on a new body. The missing female changes the input the fish receives from its group. The first visible change can look like a sex change, but it is not the whole story. A newly dominant male becomes more aggressive and takes control of the territory. That is evidence of rank ascension. It does not prove that his gonads are already mature female gonads. In a 6-month study of Amphiprion ocellaris, sex-changing fish kept male-like aggression and parental behavior for much of the transition. The authors argue that dominance behavior and sexual phenotype are not the same clock. Social status changes first. The body's reproductive machinery catches up on a different schedule. So, what carries the social information inward? Evidence from Amphiprion bisinctus shows that the brain responds before the gonad is finished. The brain communicates with the gonad through the hypothalamic, pituitary, gonadal axis, a hormone pathway linking social state, endocrine signals, and reproductive tissue. One important enzyme is aromatase. It converts androgens into estrogens. In the CASA study, the brain form of the aromatase gene, called CYP19A1B, increased toward female stages, while brain expression changed before the gonad completed its transformation. Aromatase is not a magic female switch. It is part of the chemical balance that nudges tissues toward the female pathway. Here is the turn that makes clownfish different from the usual cartoon version. A functional male does not start with an empty gonad and build an ovary from scratch. His gonad is an ovotestis. Mature testicular tissue sits alongside immature ovarian tissue. When the social transition persists, the testicular region progressively degenerates and the ovarian region proliferates. In the gonad, the aromatase gene CYP19A1A and other steroid-related genes rise toward female stages. But an enlarged ovary is not yet a fertile female. The fish still needs to produce vitellogenic oocytes, egg cells loaded with yolk, before it can function as the breeding female. The best measurements make the timing much less cinematic. In the Amphiprion bisinctus field series, gonads sampled 40 days after female removal averaged about 87% female tissue. Even at days 40 to 50, the oocytes had not begun vitellogenesis. In Amphiprion melanopus, gonads were essentially female by around day 20, but mature female function was not seen until about day 45. And in Amphiprion ocellaris, many reports took 4 to 6 months or longer to complete gonadal sex change. These are not contradictions. They are different species, conditions, and definitions of complete. The safe conclusion is weeks to months, not seconds. The group still needs a breeding male. That is why the hierarchy matters so much. The largest immature subordinate moves up into the newly vacated rank 2 position and develops as the breeding male. Smaller fish rise behind him. In the wild, this lets a group rebuild a breeding pair at the same anemone instead of sending a lone fish across a dangerous reef to find a mate. It also explains why the social queue is so strict. Every size gap protects the rank above it while keeping a successor ready below. There is one more correction to the popular story. Clownfish is a common name, not one single experiment. The 1978 histology study found the same broad protandrous pattern across 6 amphiprion species, and later work has followed Amphiprion percula, Amphiprion bisinctus, and Amphiprion ocellaris. But the exact timing and order of brain, behavior, hormones, and gonad changes can vary. In one Amphiprion bisinctus case, a male left alone after the female was removed had not begun sex change by day 35. A social vacancy is a powerful trigger, not an automatic stopwatch. The biology reads the whole group. So the accurate version is more interesting than the myth. A size-based breeding queue places one female at the top and one male beneath her. When the leader disappears, the male ascends. Social information changes the brain and endocrine state. Aromatase helps shift steroid production. The ovotestis remodels from mature testicular tissue toward ovarian tissue, and only later do mature eggs mark a functional female. The largest subordinate becomes the new male, and the anemone group has a breeding pair again. The leader's death does not flip a switch. It removes a social break, and the fish's biology rebuilds the queue.